November the 10th and
Okay, so welcome. So we're going to talk about user-friendly access to HCA and in general to ALS, plural. So we thought, so AJ and I brainstormed this a little bit. We wanted to keep it kind of open-ended because we weren't sure, frankly, how many people were going to turn up and what people wanted to discuss. So we drafted 4 slides, 4 questions that we wanted to spend 20 minutes on. There is a little bit of overlap with some of the other sessions we realize now, because the session here is kind of natural, right? But basically, the 4 questions that we have, we thought we'd dedicate about 15, 20 minutes each, depending on how, maybe some of them longer, some are shorter. What are your current favorite tools and interfaces? Let's just kind of survey the room in terms of what people are actually using and so forth. And we're going to take some notes here, right? And let's just see, use that as a kind of starting point. It's not a popularity contest, obviously. Just to think what's important, and then the obvious question: what queries do you want to be able to ask of an atlas? And we'll break that down a little bit. And then what kinds of query interfaces are needed? What already exists, and what critically do we need to create? What's missing? Right? What are the gaps? So really simple. And then how do we? This was AJ really. How do we want to manage communicating about integrated atlases where we have multimodal data, because that's something that's really fast approaching us as we sort of approach not just single cell RNA-seq data, but many different types of data, spatial and so forth, proteomics, etc. Right, with different kinds of error profiles, and different kinds of QC and so forth, and questions that might be relevant to one data modality maybe are less appropriate to another. We just need to think about that. So is this big enough? Can people see this? If you're not, you can join the Zoom if you have any visual issues and you can see this. There is this notes document that I put up on the side here. So it's a bit.ly link. So just HCA Breakout. You can go to there, and we can all crowdsource this editing. Otherwise, we have our scribe.
Sabrina in the back there.
Oh, sorry. Who is going to try and take notes in this document? Different document. How do we want to do this?
I can
Do we want to keep one document, or do you want to have a separate document? No, sorry.
I will take a separate document and we'll edit it final for the day.
Okay. Okay. Fine, no problem. So anyway, we've just sort of augmented it. We started a doc, but I'd appreciate it if people would add stuff to it. Now we'll just, the end product of this is supposed to be a useful discussion that we can send back to the group. But more important than anything else is just having a useful discussion. So we figured that we would do intros really, and we're not a large group here, so I don't think that's worth doing. So I think we've set up it. So is it okay if we just go around the room and have everybody say hi? Maybe we can all see each other. Also, we'll do the people on Zoom as well. But let's start with the people in the room. Who wants to go first? Start at the front.
Do a simple introduction?
Yeah.
Okay, sure.
Name where you are and what your interests are.
Sure, of course. So my name is Paten Ajay. I'm a assistant professor at McGill University, and my interest is actually in ethics policy and governance, especially access. I'm also on the board of directors of HCA. I'm going to be interested in hearing about some of the interests of the group, and will help us think about ways to make this more streamlined from policies.
Great.
Hi, I'm Dimitri. I'm also from McGill University, and similar research interests, and also working with the ethics working group of the HCA on how to present documentation tools. So I'm interested in.
We were just talking about that. Questions about open consent. That's great. Actually, that's something we hadn't put on there, but I think it's a really important topic about what data that is more available is data that gets more used, and I think it's an important thread for user-friendly aspects.
Hi, my name is Pablo Moreno. I work at the UBI. We maintain the single cell expression atlas resource and the human cell atlas of the Galaxy video resource with collaboration Friburg. And we are interested in understanding as well how people want to come to the atlases, what they want to find, what are the queries. And also one of the tools that we should be provisioning in our organized stuff. So
Great.
I'm Arcee. I'm the engineering lead for the CDI technology team. Obviously really interested in figuring out how to make atlases and large data resources more usable. That's kind of one of the guiding principles for the tools that we built.
Hi, I'm Isaac. I'm the lead developer on ScanPy and Pandera. I'm really interested in making sure that we can keep data points for both pilots.
Hi, I'm Jenny from Stanford, part of Lattice. I'm a data wrangler and I'm interested in data and metadata standardization.
I'm John Goshkin. I'm with S2 Genomics. We generate a lot of internal data for our own R&D purposes. We're interested in learning how to access and analyze the data without needing informatics specialists to do it for us. So democratizing access in-house capability.
Hello, I am Vinicius. I'm associate professor at the University of Chile. Currently, we recently approved a ancestry networks project to map the hidden seeds from the 6 different Latin American populations, and also to generate a map of gallbladder specific for Chile, which is very specific ancestry related. There are some diseases related to gallbladder cancer. And my task here is to know some user-friendly tools that we could implement to turn our data sets public and also to standardize some data analysis methodology of our group.
I'm Carissa. I'm part of the HCA Genomics Institute, and I'm also, our team is part of the Human Atlas Data Coordination Platform. So I'm here to learn how we're interacting with the data portal and see if there are questions and, you know, the features that people are interested in, or tools, or if there's data sets you'd like to see incorporated into the data portal. I'm here to learn about that and answer any questions.
Neil Skellmore from Harvard Medical School, part of HubMap and the data coordination center for that project. It's called the Hive for who's reading basic data validation tools. I'm also part of the coordination consortium doing similar things.
I'm Heather. I'm a kidney research group in Michigan over in the state. I have similar interests to our colleagues from the field of ethics, and I'm really interested in how we can make data sets not only more accessible, but also understandable to all patients.
Hi, I'm Becky Rainey from the same place as Heather, and I am a lead software engineer / product manager for building the KPMG Atlas tools. So just here to kind of see how other people are thinking about building atlases and how to make them differently.
Do you want to introduce something?
Oh, yeah.
I'm Sabrina. I'm working together with Chris for exams, and I'm here currently setting up the online data.
Great. Okay, Zoom people.
Max, do you want to start and unmute?
Oh, sorry, I didn't understand. Yeah, hi, I'm Max Heuster. I work at UCSC on HCA and cell browser things, which kind of overlap.
Thanks, Max. Sophia?
Yeah, my name is Sophia Telak. I'm working at the core facilities at the Medical University of Vienna, and so I'm here more like a prospective user, and also to see what kind of tools we can recommend to our users in turn, if we have this kind of atlases available.
Andreas? Andy? Maybe step away.
Yeah.
Oh, okay. All right. Okay. All right. Okay, great. Do you want to minimize that? Okay, thank you. Everyone was really good with their, I think, too. Okay, so yeah, so a good meeting is when everybody says at least a reasonable amount. Okay, so at some point, if nobody's saying anything, I'm just going to pick up. Okay, I think it's important that we sort of survey the room because, yeah. Okay, so let's start this out just as a really simple exercise, just kind of going through, and if we can add, yeah, sorry, I moved this around because I figured it was a better starting session. Let's just go through. Well, first off, let's talk about the high level sort of topics, and then sort of tools that fit under those that we all use. Okay, so cell browsers. Okay, so I'm biased. I definitely use cells.ucsc a whole bunch. So if we can add these as we're going through. But obviously, cell by gene is also fantastic. And there are many others. What are other things? What are people mostly using in the room? I'm going to pick on people. Go ahead.
Cell by gene in the past.
Yeah. And you're using that because
Cell by gene because it came out early on when I was in grad school and I'm really used to it. And it's been fantastic for sharing the collaborators now have big light plots. And it has to be like just playing that Andrea file and it works.
Right. And in using it, are you using it, do you have your own instance that you're running, or are you using the web?
For Cell by gene, I either use the website or my own instance.
Okay. All right, just curious about that. And then of course there's single cell portal over at Broad. Others that I'm missing, the BI. No one wants to volunteer and make sure that their tools group is represented.
I think today I heard about the bridge,
Is that a cell browser?
So the campus atlas is really like a subview of the EVI single cell expression atlas for mostly the human data.
Right.
But
In this context, I'm really thinking of the tool themselves rather than the broader data pool, because obviously there are sort of not trying to conflate those things together. Okay. Go ahead.
Yeah, just a very quick question. When these are mentioned, is it possible to also mention whether these are managed type access, where there's like a control system, or it's open access?
So there's two aspects to that, as I would think about it. Is the tool itself, and we're just talking about tools, open source and readily available and so forth. And I think that's true of all of those, right? Those are all open source projects. Anybody could, in theory, take them and do whatever they want with them, which is an important principle in general, in my view, for just everything that we do. In terms of whether or not all the data that they represent is open source, that's definitely not true. There are definitely managed access data sets of which we have, expression data that's sort of readily publicly available, but underlying sequencing data for reasons of re-identification is clearly not available.
Not available unless it's controlled.
Yeah, yeah, exactly. So probably available under managed access. Yeah.
I don't know if this fits in here or maybe later. I think you made a good point about, it's open source and you want to keep it in front of it. But I think there's cost, right? For many tools, this cost is prohibitive for anyone who scans their instance. And I'm wondering if this is something, I would be curious to hear, where's sort of the pain, like where's too much pain? What level would you say too much would be to set up an instance of, say, cell by gene database or the test or something like that? I'm curious if people have any thoughts.
Well, we have developers in the room, so I guess I'm curious to hear from Ruthie and from Max about the perspective. Are you getting requests for people to set it up?
It's mostly on the desktop side of things. So the platforms actually annotate these single cell data sets.
Yeah.
That's the one where we get the most requests of like, oh, can we, post it, and we'd love to like collaboratively annotate it. I think that's like one of the things that is not like currently offered in-house. You can connect it up with Cell by Gene Gateway, I think is a repo that's supported through Novartis. And that that combination allows you to list your own instance. But we haven't gotten that many requests to host like their own version of the data portal, for example. And I'm not sure why. I mean, one is there is a lot of data on there. but I don't know, I guess storage is cheap compared to the computational cost. So I think it's mostly around like the number of servers and especially for things like the expression, like the more analysis type tools, that becomes a lot more costly.
You're a hoster on the portal that you can put the data that's currently available.
I mean, you could add any data that you want, right? Yeah.
But in that model, we work the same way that it works in the desktop application, or you need to spin up separate rows per data set, or it's different?
No, you don't have to serve. They don't have to ever problem.
Okay.
Yeah, it's all the data is just listed on that page.
Yeah, our lab years, we have a cell gene instance set up just for our lab.
Yeah.
And we've got data hosted on it just so that, it's it so that everyone can get at that data, but it's not shared publicly.
Right.
Can't share that data publicly.
Yeah.
But yeah, I mean, like as a developer, I have like a 10-minute rule kind of thing. If I'm downloading something and trying to bring it up and get it working, if I don't feel like I'm making progress in 10 minutes, it's not worth my time.
Yeah, signal and the noise, right? There's so many. Yeah, I'm with you.
10 minutes. That's a strict criteria.
I mean, ish. But if I feel like it's just going nowhere after that, it's like, okay, done, moving on.
Okay, so I think it's a really good point that we do need to be thinking about developers, and we do need to be thinking about the conditions and so forth. And things to be accessible, they need to be usable from a programmatic point of view.
Want to make sure everyone on the other end can hear us. Okay.
Yeah, Max, I'm just curious. Do you have? We should keep going, but do you have people requesting to set up their own cells.ucc?
We have around 12 groups who run. Yes, the list is on the GitHub. Various, for example, the Whitehead Institute is running 40 or 50 of them internally, and you can make your own using the Galaxy single cell that Pablo created. Okay, I can see Pablo there, so Pablo can tell you more about that. If you don't even need to know a Unix command line, you can just do it on the internet. So yes.
Done by Gil and for UCSC browser on the use Galaxy and actually everyone who has data set there has some data, for instance, or the spread object, so they have tools to wrap them into the visualizers and spin them up.
I have a question. Is there a point at which you turn down those instances? Let's say you're working on a data set and you're annotating it, you want to do this collaboratively, and then you put out the publication, right? At that point, do those instances come down? Because you don't need to annotate it anymore, and maybe they're more available on like public places that you can publish once you have your publication out, or do you still continue with it?
Sorry, I didn't understand the question. Was that for me?
I was kind of
for the break. Yeah.
And so the only times that I personally set them up, they've been access control, they've been like progress, not published data yet, and then we rely on whatever downstream to us something afterwards. Yeah.
Right. Okay, I'm going to keep this going. So, all right, so that's cell browsers. So dataset search interfaces. So I think this is more at the level of high-level portals. So obviously, I'm going to volunteer the human cell atlas data coordination platforms, and we have HubMap in the room. But obviously there are lots of others. So can we add to that list EBI folks, for example?
I like EBI.
Sorry?
I like EBI one.
So that's it's technically called the Expression Atlas. Yeah. Which others are important? Go ahead.
I don't know if it's a popular opinion, but I do use Geo a lot.
Right.
Also for single cell data.
Is that as a wrangler because you're searching for data sets?
Yeah, for us, it's a really, we have, search filters that we've been using for a really long time, and that's how we bring in a lot of data from Asia and Africa and to diversify the donors in.
To what extent does Geo contain all the information that you need, and how far do you have to then?
Yeah, so it depends on how much effort someone puts into their submission to Geo, but even with a most annotated geo data set, we have like data wranglers for curators who have to read the publication, the methods paper, the methods section paper, and provide additional information that is important to single cell experiments, but is not captured in geo. They don't have a metadata schema that like optimizes for single cell experiments.
I guess that also bringing up to the expertise in the room, the managed access question also around the availability of data sets and so forth. To what extent do people feel that managed access is an issue for them? How often are people in the room actually trying to gain access to, say, the sequencing data? Does anybody ever try to get access to the sequencing data?
For digital expression data, we sequence, I mean, sorry, no, we analyze the sequence.
Right.
Sorry, like we do the mapping completely, and so what happens is our brain typically find the stuff either in Neo or in Array Express, now the biostudies, and it's normally deposited in the European Geodata Archive.
Yep.
And from there, so they grab for your answers.
So are you spending a lot of time with managed access requests?
Sorry.
Are you making managed access requests? You know, permission to
Yes, we also access the control access data from the ATA mostly and for a few data but it's very troublesome. It's very slow, right? And we try to avoid it a bit because a lot of work and takes months sometimes just to get access to the data.
People in my lab were doing sequence modeling underneath this, but I think they primarily just avoid human data at the moment because they're not like methods aren't really done.
I see. So avoid as in it calls all the questions around.
Yeah, well, access to large amounts of it, and also having to host on German servers for some portals.
GDPR and all the rest of it.
Yeah.
Yes. Great. Okay, so image browsers, which I think is sort of a hot topic. I was hoping Niels might contribute on this one. Sorry.
Was my favorite image browser?
Yeah. What do you use?
Well, I asked people in my group to use it for me. No, I think I like our tools, and then the integration of it into class or other data or Rizar. So yeah, we have to elaborate as well with whatever standard conforming your file on web server and just import from there. The browser uses this suite of tools. Yeah, so I don't want to install any software at all, and that's where I'm at. That's my heart. It's like I don't even.
And Sophia shared a comment on.
Oh, right. Yeah. Very good.
So when you say image browser, you refer to things for spatial data, for instance?
Yeah, I'm thinking of your multiplex fish, for example. We've been doing a lot of that kind of analysis, but in general, we're all going to be faced with a lot of kind of annotated image data over the coming time period in which we're presumably wishing to understand, expression within a visual context. Is there ongoing work within the CZ group on this topic?
We have a prototype out to overlay a spatial image on the Pelbay team. It's kind of hidden within a branch right now. We're not quite sure what to do with it yet. And we also kind of need to look into the scaling properties, like the resolution of the images get higher. How does the browser actually scale?
Yeah.
So a lot of interesting engineering topics there.
Yeah.
Just on the Python side, Mari is cool because you just have your analysis functions become like a button in the viewers, which is nice for when you want a person doing analysis and you're not just showing the data.
I'm not familiar with that one, so sorry. That's
Mari.
Yeah. I should be. I know it's such a growing space.
It's basically big image viewer that's all written in Python, and it's like adding plugins to it. CCI has a grant program for
cool.
paying for building plugins, and they have a lot of support from Mari.
We use a combination of the test and OpenSea Dragon This we write our applications in React, so OpenSea Drive is an easy way to display our landmark processing.
And is this doing server-side rendering, or is this all doing, is it all client?
So for OpenSea Dragon, we have to do some work to build an image pyramid, and then it just uses that.
It pulls its appropriate.
So I just got feedback from Max that he can only hear you, Benedict, but can't hear other people's questions.
Oh, that's a bummer. Let me try. I don't know
Yeah,
what.
unfortunately, there's no extension up there, but everyone can just try to speak up a little bit louder when they're talking. Should be able to pick that better.
And do you want to just introduce yourself? You snuck in there.
I'm the investigator at the Health Institute for Grant Studies.
Thanks.
I would be interested in had any opinions on like ImageJ teaching.
We don't use ImageJ or PG very much, but mainly because we're trying to build web applications. And so it's not terribly helpful for us.
Cool. And is there, in terms of the development, do people feel like this is the image space requires a lot more development versus where we are with, for example, the fairly mature cell browsers that we have? I'm guessing the question answer is yes, but I mean, there's been a lot of work in HubMap. I mean, it's quite impressive.
I think so. What I guess, yeah, the question for me is, I think, like, so the image browser is like one spatial modality, essentially, right? I think, but we can look at images. That's not so it's really the problem, but it's more, how do we integrate all the other images? Like, how do we go from 3D images to 3D? I think that's really where the challenge lies going forward.
Yeah.
And then I think there's another dimension that's sort of like, I mean, I was surprised that Napari is actually a desktop application rather than something that's web-based and next to backend. I think that's where we're kind of, there's nothing really there right now that does analytics and then displays the results or allows you to interact with the analytics in a browser based. That's I think there's a gap there. I'm not, maybe there is something out there that but there seems to be sort of a separation of now desktop tools to do analysis and then sort of viewers that are browser based.
Is that because of requirements around GPU usage or general access to graphics?
I don't know.
I think a lot of it is that it's two languages, and the person who writes the analysis code might not be such a great JavaScript interface developer.
Right.
I'm certain.
Well, kind of adding on to that, I'm really interested to understand, and it's actually kind of jumping into the next question, like, it's an extension of the question, but what are the queries that we want to make of the spatial data, like interplay with the like RNA sequencing like data, right?
Yes. Yeah, I think let's hold that and let's get to that in a minute. But yes, I think that's we should keep going. All right, let's talk just briefly programming frameworks. So yeah, so obviously in Python. Yeah, right. and obviously in R and so forth, the whole semantics, etc. Are there other frameworks? I mean, do we feel like that's where we've converged? That's a fine answer, I think.
Well, you have the one from frameworks, right? Like React or Vue or.
Right, but those are more general in some senses.
But I mean, most of these visualizations is there, right? Like intense is JavaScript based on something, right?
Right, but it's JavaScript, but then you can use it both tomorrow, right? So,
right. Okay, I'm on the time, so I think we should move on. Okay. All right, we don't have to go. We put up a bunch of questions. We can talk through whatever's interesting. We don't have to go through this immediately. So yes, going back to this question of so let's just briefly go and try and survey the room about what queries we want to be able to ask of an atlas. So, all right, we put up a bunch of stuff here. What's missing? Okay. Can everybody read this just about? Okay. So where is a given gene or protein expressed? Let's say you want. I'm a biologist. I'm studying a given particular gene, and I want to know about where it lives in terms of my atlas. Okay. How do I search and browse by cell type? Right. Okay. I'm really interested in this particular cell kind of lung cell, and I want to look at it I want to see images. I want to see all the data that you have about this particular cell type. Okay. How do I classify a new cell or image given the atlas, right? How do I bring my data into just sort of an analysis question, but it is an important question, right? How do I annotate my images? How can I just get access to raw sequencing data? Do we have decent programming APIs for that? For a given disease or specific condition that I'm interested in, how does it relate to the given atlas, right? Which is a very general question, but something that I think people, are struggling with. And then this was your question. What is an atlas query really? Like, is there a sort of generic set of questions or covering set of questions that we can come up with that basically, map the space in terms of the primitives that we need? Okay, so that was our starting point. What do people want to add to that?
And I have a clarifying question.
Yeah, of course.
First question, and you define, what do you mean by where? And what do you mean by expressed? I think we can answer that. I think we can build our basis.
I think for us, one of the basic questions is, how do our data sets compare with what's in the atlas?
Right.
Is our data set grounded in reality? Are there artifacts that we're seeing? If we see differences, then what are they? And are they statistically relevant? And what do they mean? And, but I don't want to go down the rabbit hole of analyzing some difference that we see with our own data set. If it's, an artifact or some function of an experimental parameter that's not really relevant to the underlying disease.
Yes.
How do you expect to do that query? I mean, do you expect to compare that directly based on an expression profile matrix, which would be like a very heavy top down, or do you expect to compare that by metadata, simply saying my data sets are about this disease, and I want to see how do you?
Yeah, so I don't have any expectations because we're new to this, and I'm very naive to it all. So, but those are the kinds of questions. As a quantitative scientist, as opposed to somebody who's used to looking at just phenomenology, I want to know. I've got four data. I've got data from four different patients. I've got matched normal and tumor. So, how statistically relevant are the differences between the normal and the disease state? Do my data sets compare, or how do they compare with what's already in the analysis? Do I see the cell types that I expect? Do I see the same kinds of differences that people have already seen for small cell lung cancer? Are those data consistent? Do I have enough statistical power in my data to know that those differences are real? Do I need to aggregate my data sets in order to build a statistical model? So those are the kinds of questions that I want to know, just about my data any quote unquote good, how they compare with what's already out there. So I don't want to waste time investigating something that's already known. So if we see anything new, see anything that's interesting, and I don't know how to do that. And I heard some fantastic thoughts this morning. I mean, they're incredibly helpful to nobody what's out there. But I also heard a lot of things that are in progress, things that are in development, things that will come out next year. And it's not worth my time to figure out which platform is most useful for asking a certain kind of question. And so having that kind of guidance is incredibly powerful. Oh, these are your data sets. These are the questions you're interested in. This is a data flow or an analysis flow that with the tools we have today would be most useful. And I don't see any of that anywhere.
Yeah, I think that's a hard question because it really depends on what your skill set is.
So it was easy.
Yeah.
As a naive person coming in.
Yeah, no, I
really have to go out and learn the details of half a dozen different platforms that people are developing, as opposed to, oh, you're looking at these types of data. This is really the best one right now.
Right.
Best curated data sets or whatever it might be.
Yeah.
And right now, that's the position that we're in. It's naive users just having to sort of learn about everything because there's no guidance on.
Right.
I think there's also, we don't really know what the best way to do things are yet, so it's hard to get that.
Yeah,
Right. And
yeah, I understand it's what it is, but.
Right, if you have 5 different people, which is the best, they're gonna give you 5 different answers,
Yeah.
at least.
Yeah. Sure.
There's also a comment, Sophia.
We also sometimes ask ourselves when performing a new type of experiment whether it worked all right. So, e.g., if a liver sample looks like an average liver sample,
Yeah, it's a great,
how do I choose?
great example of what.
I think another kind of broader group of questions that I don't see up there is just, what does the clinical data look like in this cohort? And how can I make my own cohorts given the clinical data that are available so I can figure out like, hey, is this more prevalent in men versus women or smokers versus non-smokers or whatever?
Yeah, so I mean, I had two things. I had a plea for kind of documentation and guidance and a kind of higher level kind of, this is like all the way from the sort of pedagogy of how you actually learn this area, right, through to having something that holds your hand as you go to, oh, I've got this data, how do I analyze it? And then, yes, right, there are very, very specific questions about how you apply them to your given data set. So do people feel that training, I mean, it is difficult, right? If you feel like everything's under development, it's really hard to like, this is what you should do because you feel like abroad, you don't really know yourself, right? In a sense, that's half the question.
So that training is a huge problem. And one of the reasons is, you guys who know this stuff are incredibly valuable and incredibly rare. And it's very difficult for an organization like us to get access to people who know this field. Can't just go out and hire a bioinformaticist. I mean, it's really not easy to find people.
So are you suggesting the HCA should sort of create more training and outreach?
Yeah, exactly. And so if we had more resources to train people up, and I have people who know Python and know R. They're the generation that grew up, learning that stuff. I used cars when I went to camps. This is not the world I live in. So yeah, if there were, and maybe those things exist, maybe those resources exist, and just we're not aware of them. But that would be a huge benefit.
Yeah, I would, as a comment, as an academic who teaches intro programming to, well, I guess collectively we teach about 2,000 undergrads a year how to code in Python. That's about 20% of our undergraduate intern and 30% of our undergraduate intern. It's a completely different world in terms of, if you were in school 10 years ago in biology, you didn't do programming, you do now, right? And I do think that there are far more people who are at least semi-computer literate, programming literate, who are coming to this as sort of people who potentially can use Python, potentially use R, but don't have any clue about this space whatsoever, right? So yeah, I agree with you. It's not just naive biologists who want to click on web browsers anymore. I think we have this growing base of people who are going to be capable of using tools if they know what questions to ask. Good. Okay. So yeah, I agree with you. I'm wondering at some point we should try and boil this down to a set of recommendations and stuff. So maybe we can try and work that in at the end.
I wonder from that perspective, whether some of the first order tools that are coming out there will be useful for someone like yourself, like Azimuth, for example, is basically a label transfer tool.
Yeah.
Where you just have to have a solid key matrix to go into it, reference, a well-annotated reference that you're mapping against. Then you could have label transfer accuracy as a metric.
That's a great example. So we run Cell Ranger, and we've got a bioinformatics consultant who got our data into AWS and set up a pipeline to go through Azimuth Saran. And so we can do that. But as an example of some of the limitations, what's the right reference to use for the data set? We did sequencing on TILs out of a lung. And if you use normal lung tissue as your reference, you get a certain set of annotation. And if you use, a different reference set, you get a different set of annotation. And which genes do you pick as your canonical ones? There's a lot of subjectivity that built into that. Also, you don't know when we're putting garbage in. So So that exists, no, do it, but it makes it's real nice.
And for the people developing tools in the room, I mean, do you have other questions that you get asked every day that are not covered here or that you want to add?
I wonder how people are doing normally their self-typing. So I noticed it happen, right? Either people go with their matrix and they try to sort of use a convoluted method that will based on the expression itself type cells. Or I see also research that you just take the top 10, 20 markings for the cluster and they go to some service and sort of get an idea of what's self-typing. What do you see in general regarding, I mean, or what would be the queries for self-typing that you would like to be able to do?
How do you identify cell type by
With the
sort of,
data,
yeah.
like from the expression matrix or like from the just sets of genes or some other approach, I don't know.
Anyone want to respond on that?
I think people should count the expression matrix, not just the set genes, but that's more personal opinion.
You're right.
But do you know if there's evidence that the assignment is a lot better compared to the actual markings, for instance?
So actually, I think this is something that really needs stronger benchmarking, is the quality of how well we can actually assign cell types, how well it's like, I'm not aware of really strong benchmark response methods.
And as far as I understand, it gets even more complicated when you get into disease tissue, because the cell expression is very different in disease tissue versus a healthy tissue. So, looking at gene expression, you might not be able to tell that that was a podocyte or whatever. It's going to look very different.
Right. Particularly if you take your top 10 genes, right? That is definitely limited.
I have a vague understanding that this is a bit less of a problem within industry because they will be able to generate it's like similar data sets many times. They always generate the same way, but a lot less like technical match effects. Then it becomes easier to see like how versus disease. We also have had many more in-house samples to look at. So I think like publicly we have as well annotated data sets that you could train a good classifier on. Right. I think. Yeah.
It seems to be one of the guiding reasons for the open problems, the benchmarking effort, right? Is to get as much broad set of data as possible, run these benchmarks on it, on these various algorithms to try and get some results, be able to have a good understanding of what method might be right for your data.
Right. Good. In front of what time did it go until?
We can go till 12, lunch at 12:45.
12:45. Okay, and we should also have a break. Okay.
I think you're going to easily expand on for a given disease. It's not just gene expression, proportional differences, it's also
Right. Yes. You mean?
For example, in neurodegenerative disease, you might see loss of particular kinds of cell vulnerability. So proportional differences are a good thing that lots of people are pursuing it. And not the reference to changes associated with
Do you think that this is something that lives in a public database? Is maybe just a collection method enriches or does not apply to itself because character cells character will have a collection database?
Well, I mean, there can be technical reasons as well as biological reasons, of course. But even just getting that read on, what you show where you should get them for one or the other.
Yeah, I think it's probably less complex. You can actually see the cells were there or not, but with microarrays, it's a bit more complex.
With spatial data, you end up like the cells end up there's deterioration and like this weird stuff happens. They end up being misshapen and things like that, which then makes it hard to go, oh, that's on one of these cell types because all of a sudden it just looks really different. So you get scarring and all kinds of stuff in there, which makes it really hard to tell what it was.
I think also the spatial data at the moment is still pretty sparse. You spend $3,000 on a tissue section, and there's your data set where it's getting much better coverage with the same cell for single liquid. So until that rate changes, you're able to do lots of serial sections, build up a big data set. I think it's pretty sparse. So anyone who does histology, can compare it a lot.
AJ, is that going to change in PubMed this next round? Are we going to be drowning in image data? Say yes.
Probably, we'll have a lot more.
Good.
We're doing less experimental technology, so.
Right.
I think to come back to one comment, I think the one on the chat here about, like average liver, I guess it's in quotes, but I think to, like, how would you answer this? Like by looking at the distribution of cell types, or I don't know. And then if you look at that, then the question is like, where in the liver did you take that sample from? Because they have the whole liver, right? And I think that really is one piece to it. I'm not aware of any query interface right now that will allow you to choose like a location and then say, like, give me all the data sets in that location. Most likely they can send to build anyways because the data is so sparse right now. I can look at chemical distribution of samples. But I think that's where we need to go also, right? To answer these questions. Like, when I take, I think if there are samples taken from a very specific spot, right? So maybe at that point you could start asking questions about what is a typical thing look like. But then you also disease, where things have occurred.
And the progression of disease, like a person might come in for a healthy tissue, but they might not really be healthy. They may be somewhere on that spectrum toward disease. And so it's hard to say that, are they really healthy or are they actually like going toward getting disease?
Yeah.
So in single expert atlas, we record the sampling site.
Right.
But we do find for very few studies in general, so it's not widely available for them. And it's searchable, but again, it's fine for it.
But it's searchable, but then once you've got that site, then it requires a bunch of manual kind of analysis to look at, to determine.
No, I mean, essentially, if you hit the jackpot and you search for a sample site that actually is in the index, you will get a couple of data sets that sort of have that sample site among its metadata.
Right.
And then you go from there. So
one of the basic problems is are the data coming from cells or are they coming from nuclei? And just right there, you're going to have a substantial change in transcriptome from the state. Right. And to know what the right references are comparing to. Right.
Yes, the multimodal data question. Okay, I think we should keep going. Just trying. Okay, so that was good. That was useful. So then our third question is sort of follow on. So, what kinds of query interfaces are needed? What already exists, and what do we need to create? So it's very related to what we just discussed. So I mean, in the HCA GCP right now, we've really focused on kind of faceted search, so subset search of of the metadata. And that has been a difficult nut to crack, to try to get people to be consistent about the descriptions and data sets and so forth, right? Just solving that one problem is hard. Within HubMap, there's been a lot of work, I think, on much more work on looking at making images sort of viewable, browserable. But do we have, what about image search, and how do we see that kind of developing? Questions around sort of free text search and to what extent natural language processing, publications, mining of actual stuff that we write as academics, and so forth. Right? How important is that to us? AJ, you brought this in. Everyone's just using Google to search the data. Is that a reflection of the fact Google's great, or are we doing something wrong? And then, yes, I guess going back to the question of spatial results, but this was an incomplete list. So maybe we should just start with what are the things that what are the kinds of interfaces that you all think are important or that you're getting requests for?
I'm gonna have a comment, which I've been dragging a question that was in the previous slide back into this slide.
Yeah, sure.
Because one of the anomalies that I saw in the previous set of questions is how can I download a cell-by-gene matrix?
Right.
And a lot of these questions and almost everything that we've been talking about is more towards kind of the hosted side of things, where everything is on the web.
Right.
And so this brings up a question to me, like, why do you want to download it? Like, what queries are you trying to do with the downloaded data that you feel like it's not enabled by the hosted tools?
Well, don't you? So, Linka, I mean, don't you oftentimes that the matrix is not very large? You download it, then you throw it into your programming framework because you want to ask you know, you're a programmer and you want to ask very specific statistical questions that are hard to do.
Yeah.
Is that?
That's very good.
And also train a model on it, concatenate things together and combine them.
We want to look at them in a different visualized.
Or compare data sets from different places.
Often I want to combine my data. Which maybe you don't want to upload it to a server, maybe you also want to use some methods and upload it to a server. Yeah.
Yeah, I agree with that. And I think we just as a comment within HCA, we've always tried to make the data sets exportable within into Terra, which is, you know, the Broad's platform. And that from there you can launch an open service. You don't necessarily have to download it. You can. There are lots of hosted services currently as well, so for running things. Yeah, but in terms of the interfaces, what are people trying to cook? What are things people are cooking up in their labs, right? What's the next? Assuming that we don't believe we've got to a perfect state, I definitely don't. We've got to a perfect state.
Here's the work that David and Spyro is doing and talking with Cell by Gene about just like actually having consistent metadata so you know what fields are going to be there.
Yeah.
And I think that's huge, just like having a bunch of data sets with consistent metadata.
So just ensuring that the data is wrangled so that we actually have everything well described. I mean, that should be a function of HCA. I mean, that's what the data explanation platform is spending a lot of time trying to do.
So I think I attended the Amnesty Network meeting, or there was a meeting earlier this year, and one of the topics, or like one of the breakout sessions, there was a conversation where in addition to like the metadata that you can query different portals and resources, like need like very specialized language for ancestry research, and I don't think that's currently available to the extent of the community anywhere. So I know there's just like very community based that I think.
So you're talking about the question of when you have a data set that's derived from an individual of a certain, you know, genetic subpopulation who wants to make sure that if they're doing cross comparison, that there's well controlled. That, for example, they could find other data sets from other individuals of that same subpopulation. Yes.
Yeah, I think that is supposed to just having good ontologies for a lot of stuff. I mean, I know that kidney, there's a lot of different terms that are thrown around to mean the same thing, and cancer it's the same way. Like defining an ontology that can map like all these 5 terms to one, so that you can search on any of them and find the one thing, so that you can actually do that comparison and know that you're talking about the same thing, or you're talking about subclass of that thing. So you kind of understand that, and really get more value out of it.
Yes. Would you want to comment on that, Teresa? I mean, how much within the metadata?
Yeah, ontology is wonderful. Helps keep things standardized, but also like, I guess like interpretation of, I'm like using the ancestry meeting just because I have an example, but you know, there's like self-identifying ways and there's like grades, so just like making sure all these researchers are speaking the same language before you start to ontologize it. Yeah. That was like one of the topics that came up. So if we're gonna, you know, query and like look up race, like is everybody referring to race? Right. Just like,
yeah, we could go that's a rabbit hole that we don't have time to go to. Yeah, we couldn't go down.
I wonder how other resources are treating the free text search problem. I mean, because on one side you have your ontologies that have certain theorems and certain relations in terms, but on the other hand you have the user that have some free text, and of course there's also where you can sort of lemonize for rules and so on. But I mean, we do that for instance to Sonar and Lucene, but I wonder if how other resources are sort of taking care of getting from the free text to the ontology terms when people search metadata.
It's a good question. Anyone want to comment? Max, do you want to comment on that at all? And you've done a lot of work in NLP.
I didn't understand the question, sorry.
Well, in summary, the question was, how does one link free text to, for example, the metadata and so forth that's contained within a given sample and so forth, so that it's discoverable, so that someone could actually, ask a natural language question and get back data sets and so forth. Is that summary?
Yeah, or even how you actually get to the ontology there, because the free text search will never be essentially matching exactly what
Right.
it already has.
You're not necessarily aware of the controlled vocabulary.
Sure.
If there's a controlled vocabulary associated with an ontology that somebody does not necessarily using.
Exactly.
So the DCP has full text search, right? And there's an abstract for every data set. So you would ideally find that. What else? NLP on dataset descriptions is not something that any database that I know does in biology, like a structured parsing of like grammar parsing, finding words and nouns and replacing. If there is an ontology, yes, that would work. But what types of fields? I mean, then we'd have to discuss. Mostly, it's the organ, and I mean, cell by gene and the cell. The new CCI single cell portal does this, right? Especially for organ. There is CCI has an ontology for the different organs, and you can see the structure. And if you check brain, then all the subtypes of the brain get automatically searched for. The CCI website also has full text search, as I think, but.
Searching abstracts, then or searching the paper associated with it, that's putting you on the spot.
I think we have free text.
Oh.
There's a disagreement that
It doesn't have full text search. Okay, sorry, never mind. Okay, so maybe it's the DCP portal. But otherwise, if there's a paper associated with, that's not something any database does, but it would be nice to have something like this. Yes, maybe. I don't know. It's just far from. There's just so many things to do right now that it's never been a priority to data mine the paper associated to it with a data set. It would be almost a separate project. It would be very nice, but I don't think we are there.
I think this would be a really good question for Evan, because he said that they were doing this for their ontology search in his talk earlier today. But he's probably at the metadata section.
Okay, yeah, we need that. Good. Cool. One more thing before we move on to this. I mean, for a long time, I've talked to people who wanted to provide a blast-like search for cells and so forth, right? So that one can come along with a set of marker genes or a vector or a matrix in general and be able to find and so forth. To what extent do we feel does that exist at this point?
There's a tool called Cell Blast.
Okay, yeah, I figured it exists.
I haven't tried it.
Haven't tried it. So that's not something that people are wanting to do day in, day out at this point.
I mean, so just like search, upload expression, get back something.
Well, within the DCP, for example, right now, we don't have a way of searching for data sets that contain a given gene that's being expressed at cell levels. There's a whole bunch of like gene-driven or expression-driven questions that they're not serving. So I'm curious about.
HubMap has this search where it can provide a marker, protein or gene, and ask, like set a cutoff for expression. That's why I asked the question, what do you mean by express?
Yeah,
I don't believe that means.
Right, yeah.
And then,
give
me all the data sets that contain cells that express a certain level or above a certain level. If it's at a minimum number of cells that you want to find in that data set.
Yeah.
And then you get a bunch of data sets back. So I guess you could imagine extending that a little bit and saying, well, we want a combination of cells expressed.
Mm-hmm.
Or sorry, combination of genes expressed in cells and get the data set. So I guess if you were to merge all the data sets, you would get a bunch of cells back somehow. And these cells would then be associated with locations, either in image or in anatomical, like a sample locations, like that. So, yeah, this is sort of a question where we've been trying to, it's been implemented.
Right.
But it's not.
But a lot of people using it, I guess.
Well, it's implemented, it's not really released because there were a lot of challenges. We're not involved back in implementation, but we're UI developers back interface with the API.
Yeah.
That was slow.
But you intend to ask the user both the GitHub for a level of it.
Right. And so this level of expression, for example, is really hard for me. what do I put in? You know, what is, what?
It will be interesting to see when you put it out.
Right.
And have some analytics to
Exactly.
to see whether people actually use the other box to say something or they just use the keys.
But I think this is indeed one of the challenges with this approach. It's like studying cutoff is really hard. I mean, if you start playing around with it, after a while, you might get a feeling for that particular marker in the data set.
So what we do with that is that we get an insert, we essentially just give the user all the data sets where we find that, but we rank them with data sets in higher positions when the gene is a marker in one of the set types of plastic.
Yeah.
So you have all of them, but then at the top where it's marked.
Yeah, that's great. So yeah, I guess then the question is like, what do you want to do? You just want to know where it's being expressed. Yeah, indirectly, you get, I mean, you get, in our case, you get data sets, and the data sets are annotated for you. Right, so you could sort of see, okay, this gene is often expressed in the kidney or whatever, if you get 90% of all kidney data sets. And maybe it's expressed in some other tissue as well, or could see something at that level. That might be sort of a more global insight rather than just here's a data set. Right. Yeah.
Okay. Yeah. No, I can see a lot of things that I'd like to do, but I don't know if anybody would actually want to do them. As a developer, I can imagine creating it within CCI. You try within. So, what are you trying to create kind of gene-driven, or do you get demand for questions from people?
Yeah, so we just did launch a new tool. It's about the SD expression tool. So you input a set of genes, and then you input tissues, and it gives you kind of like a dot plot of where that gene is expressed across the 12 types of the tissue.
For a given data set or for a full data set?
Across all data sets.
Okay.
But you can also filter down so that you can like exclude certain data sets or just pick certain data sets that you're interested in. And it also allows you to compare against the standardized metadata that we have. So ethnicities or the.
And the units for the expression going, are they TPMs or are they like Z-scored, like normalized from some?
Yeah, so we use Rankit to normalize the.
It's like I think
It's like an. Yeah.
I think it would be very, just brainstorming here, but I think it would be very useful to think about how to integrate that within the DCP. It's something that people keep saying, well, yeah, you provide all this great sort of search over the metadata, but what do I do if I want to come at it from a gene-centric point of view? And I think, yeah. But in general, I want to see much more integration between cell by gene and the HCA DCP. There's an ongoing discussion of making sure that all HCA data sets have all the metadata associated required for cell by gene, because you have a rather opinionated. In a good way.
Schema. Yeah,
Yes,
I think
exactly.
we're mainly on trying to keep our schemas as minimal as possible,
Right.
but just enough so that you can kind of do the downstream analysis.
Right, but you do require, for example, having the cell annotation across data set, which is something we have, right?
Cell browser, no, I mean, we don't require the ontologies. That's the, you guys.
The ontology of cell types.
Yeah,
Right.
exactly.
Yes. Okay. Yeah, I would like, anyway, separate to this, I would like to follow up and make sure that we are thinking about that the next round of DCP. Yeah. Cool. Okay, we are running out of time. I want to give people, we don't need to go in, but we had one more question, which goes to this question of integration. I think it's important. So yeah, so increasingly, we're all being asked to think about multimodal data sets of all different stripes and colors, right? So you're going to have proteomics data, you're going to have, different kinds of single cell, many different kinds of single cell data, and possibly also image data to boot and so forth. And investigators will go nuts and create every kind of data type you can imagine. So how do we think about making this easy to use, right? It's a really tough question, but have people got thoughts? I mean, we lost our Alan Brain out, but for people who are trying to build analysis, people have thoughts about what's the path here for trying to make it so that people can bring data sets together, compute on them, and also understand the limitations as well. Question. I mean, from the Scanpy point of view, you must have people you're doing a lot of different kinds of development. I don't know how much this affects you in your everyday life, but are you getting questions about how to integrate different kinds of data together? For sure.
But I'm also thinking about like when people say like integrated data sets together, I think there's
What does that
different
mean?
use cases.
Yeah, what does integrated mean?
Well, but it's
Yeah.
also like there are a paper comes out that integrates a bunch of data sets, and then that's a data set who want to download, or they want to use a classifier that's been trained on that.
Right.
Um,
But is it all just one big latent space that you want to represent where everything's just been kind of boiled down to? And what does integration mean, I guess?
well, I think like, do people really care about getting that one big latent, downloading that one big latent space, or would they prefer to get a classifier that's trained on this and then use on their data?
Mhm.
I think most people actually want their classifier.
The classifier, so what does the classifier just be? Let's get a little less abstract. What does the classifier allow us to do once you trained it?
Ask where my data sits relative to this data.
I see. So it's essentially build an atlas, which is build the space, and then the classifier of that space. So then, where do I sit within this universe?
Yeah, I mean, I think there's also like whether it's projection or categorical classification, or like if people want the categorical, but in more cases, people want the just tell me what cell type this is, tell me what state it's in.
So do you,
Less than where it sits in the graph.
right. So in the long run, is the HCA one big machine learning model whereby we're just like, okay, all right, I've got this new piece of information. Where does it sit in the universe? Our knowledge of cell biology. Yeah. Okay. Good. I like that answer. Good. Yeah. At some point, if we have just one nice big machine learning model, our life will be waste. Yeah. Does anyone have experience doing, for example, proteomics and singles and different kinds of single cell genomics and how they're integrating that together? The challenges with that. I'm curious, Niles, from your point of view from PubMed. You're displaying a lot of different kinds of data.
We don't have, to my knowledge, we don't have any data sets where we would have like gene expression, transcript level protein, right? That's the same. Yeah. So I don't think there's any integration. I mean, I have.
Yeah, I guess this comes down to the question of building an atlas.
There are data from proximal slices. So we get, we get codecs and RNA-seq.
Those kits are coming.
Yeah, so
A lot of sites we see.
Yeah. I guess, I mean, like the first part of the question, right? I think our gene code is really very similar. I mean, if you were to, well, so what assumption are you making? The assumption that should they be the same? I mean, maybe they shouldn't actually. So I think that would be, at the minimum, would be helpful to have a way to see, what do I retrieve when I look for, say, is this protein present or transcript present? Like just being able to compare that. On about the same tissue roughly, right? Again, it's like we're looking at is it what part of the protein that being able to just compare relative to the same cell types, for example. If not, what are the differences? But I think if you're interested in this is biological or the assay, I think being able to drill into the actual data and see the molecular level would be very helpful. Not only abstract data from cells, probably, so at least for now, until we have a better understanding, it's really cool.
So one thing that I have been, I think I've heard 3 or 4 people separately propose this idea of having just better gene regulatory information available that we can then integrate. And like maybe you have done some multimodal assays, but also maybe you just want to go and look at results from other multimodal assays and try to use that regulatory information on your data.
When you say gene regulatory, you're thinking sort of network information, so priors about, okay, this is on, this other thing should be on or up or whatever.
Yeah, nothing based on like taxi.
Right. Yeah. That's a very good point. Okay, I want to, we're basically at time. So and I also want to chat with AJ and Cliff to summarize this all together. Does anybody say something like that? I don't know. We all have a lot of thoughts. No, we'll synthesize. Thank you, everybody. This was great. It was useful for me, at least. I learned a lot. And I think we can say some useful things. Does anyone have any final comments or thoughts that we didn't cover today that they're burning to talk about? I think we did our job. That's good.
I mean, I guess the only thing that I would say is one thing that we do when we're developing our tools, to try and figure out who we're developing tools for.
Yes.
So make sure that you know who your personas are.
Yep.
And develop the tools for that persona.
Yeah.
And
HCA has tried to do that. I've always been a bit of a skeptic because I feel like the persona is changing. Like people assume that bench biologists have no ability to script. Sure. But I don't think that's even true anymore. Like, oh, go take a Python course, and then oh yeah, they come back and they can use Jupyter.
right.
So I do feel like it's
You do have to
evolving.
understand who your personas are and kind of
Right.
actually talk to some people who might be users and try and like define them based on kind of what people are now, not on preconceived notions.
Yeah. Really great.
And I think that's, I was trying to make that point in my presentation, because these personas, the use cases that we're talking about here, in the context of HCA, every single app project has the same personas, the same use cases, really. I mean, there might be some differences in use cases, simply because certain data might be available. But I think that's really something that, you know, there should be essentially one paper that says, Salad is here are the people who want to interact with them, and here's what they try to accomplish with this information. Today, you know, it might change in 5 years. Like, you know, if you would have asked 5 years ago, there was no spatial data. Like today, spatial data is a hot topic, and we need probably use cases for that. But I think that's something where the community will come together. And I don't want to say solve the problem once and for all, but at least, you know, for all the tools that are out there, have sort of almost like a checklist, and that would also help us compare tools better to each other. Because
Didn't HCA try to do this at some point?
Yeah, I think this goes back to what John was saying, like as like someone who's new to this community, like today, even as someone who's been attending these like meetings since 2017, I learned about so many different portals and tools, and I was like trying to in my head create a spreadsheet where I'm like, okay, they do this, but like if I'm having trouble, I can imagine for someone who's new, it's like information overload, just having to navigate is funny a lot of times. So I think one of my main takeaways from this session.
So the use case thing is really interesting. We've gone to commercial companies that are trying to simplify things and create user interfaces on top of tools. And we think, at least for us, they completely missed the mark.
Yeah.
You know, and they haven't been helpful at all. So it hasn't been the solution that we thought it would be. They just go out and buy it. You really can't.
I mean, you have to do iterative design. So like you go say, okay, these are my people that I'm trying to target. Do the thing, and then I have to go back to the users and be like, did I hit the mark? And if I did, then I gotta go back and make some modifications so that the tools that we're building are actually usable. Not the word.
I do think it would be good if people put out more of their analytics, right? We know who's using the HCA DCP and which data sets they're downloading and so forth. And it would be, and I'm guessing that for some of the programming frameworks, at least know who's downloaded and so forth. No?
You know how many?
You know how many, but not of the specific. Yeah. Agreed.
I think I'm effort around that.
Okay. We'll work that in. All right, we'll work that into our 5-minute comment. Yeah. Great. Yeah. Any final comments from Zoom? Thanks everybody who participated. All right. Well, thank you.